Thanks very much. Karim Lalji from Microbion Corporation. I'm very excited to actually share with you some information on our company and our programs under development. Skip the forward-looking statement. I'm sure you can read that very quickly. We basically are developing something we believe is very unique at Microbion. We have a new antibiotic class. We've actually received that designation from the WHO. We believe this is the first new class that's been designated as such by the WHO in the last 20 to 25 years. And what is unique about the approach that we're taking towards managing infections is that not only do we eradicate the bacteria, we also eradicate the biofilms associated with that bacteria. And we believe that is a unique differentiator for how we're achieving our efficacy. We also are taking a unique approach towards development. As many of you may know, the market capitalizations of antibiotic companies are not faring that well, definitely not in the United States, and I believe it's the same here in Europe. And I think in large part the concern is that when you develop a very potent, broad-spectrum systemic antibiotic, it may be put on the shelf and reserved for only when that product is needed. And rightfully so for many reasons, but that creates a challenge between the commercial needs of a company and the public health, perhaps constraints of trying to reserve therapy. So, very early on in our development, we wanted to focus on areas that would not result in us falling into that trap. So as such, we have taken towards a targeted delivery approach for our broad-spectrum product, which we believe actually facilitates antibiotic stewardship. We also are focusing on unique clinical endpoints, which therefore makes the product less substitutable by generics, because there aren't any other products, for example, that have that indication per se. And we also are focusing on strategic indications which would not necessarily be hospital-based indications, which becomes a bit more problematic from an antibiotic perspective. And so I just wanted to elaborate a little bit more on that statement. We have 3 advanced programs, or relatively advanced programs. Our first is a topical treatment for diabetic foot ulcer infections. We have just recently completed a phase 1b study in DFI, and we got some very interesting signals of efficacy from that study. So at that point, we decided to go to the FDA. We had our FDA meeting just last week in Washington, or just outside Washington, and we have a very clear pathway forward with the acceptance of a unique indication, or unique endpoint, that we believe would really be quite valuable for us as a company to move forward with. And obviously, it's provided a lot of de-risking of the regulatory risk by knowing where to go. Because what we did see in our program was a reduction, a pretty significant reduction in wound size. Now, these aren't statistically significant. It was a small study, but it was directionally significant, which allowed us to open up a dialogue with the FDA. So we are actually now looking to do a definitive proof of concept study as our next study in the diabetic foot ulcer infection space. And in our discussions with the FDA, if we size it appropriately, they would accept that as a pivotal study, which is actually quite interesting. And it's a topical formulation. And also what's very interesting is that when you're dealing with chronic wounds, they are known to be both infected with bacteria as well as biofilm. And I'll spend a little bit more time on that in a few minutes. We also have a program that just completed in a phase 1b/2a program in orthopedic infections. And the top-line data has just come in, and we expect to have that final data in the next month or two. But we're very encouraged by the preliminary results in terms of the safety of the product, and some insights that we're also receiving on the efficacy of that product, even though it's a very small study. And we also have a program in cystic fibrosis-related infections, and I'll spend some time in a few minutes providing more detail on that program. as I mentioned, we are a potent antibiotic. We are potent against both gram-positive and gram-negative bacteria. And we work against MRSA, VRE, the multi-drug resistant gram-negative bacteria, and also all of the escape pathogens identified. So it's a very broad spectrum agent. Our activity in the bacterial space is separate and independent of the activity in the biofilm space. And then again, we also have activity against a broad spectrum of multi-species biofilm, which is part of the mode of action. And we also are broad enough in our spectrum that we also work against fungal fungus as well, but we don't actually have an active program in that area to date. Basically, our first or our lead program is in DFI, and biofilms are known to be present in the majority of patients infected with diabetic foot ulcer infections. And they're also known to be polymicrobic, meaning there's multiple pathogens that are involved in the infections. So it's very important to have a broad spectrum of coverage, not only on the not only against the bacteria, but to also have biofilm, anti-biofilm activity, and we achieve both. So we believe that's a very important starting point to be able to address the biofilm and the bacteria, and the broad spectrum of the bacteria as well. What we know based on data in the United States is that there are approximately 100,000 hospitalizations on an annual basis based on diabetic foot ulcer infections. We also know that there are about 70,000 amputations on an annual basis in the US based on diabetic foot ulcer infections. We also know that there's a mortality rate which is about 50% within 3 years of amputation, and it's about 70% within 5 years of amputation. So these are really very serious, they have very serious downstream consequences if you actually have one of these infections. And we also know that the biofilms present are very broad spectrum in their own rights as well. So therefore, we believe this was a classic type of population to test the hypothesis of our ability to treat both the infection and the biofilm. I won't get into this in detail, but there's also a lot of data in the literature which indicates that the presence of biofilms impedes the healing process, which is why it's important to treat the biofilms as well. I just want to share, very briefly some information on the trial that we just completed. It was a randomized double-blind study in moderate to severe diabetic foot ulcer infections. And we targeted chronic wounds. We did not go after acute wounds. We wanted the most difficult wounds, the ones that really don't heal, to ensure that we would be able to really identify whether we're working or not in the most difficult infections. And it was a dose-ranging study, and we basically were looking at both not only safety, but also trying to see if we could get signals of efficacy as well. And what we did find is that the drug was well tolerated at all doses, and we had a prolonged median wound size reduction signal, which was, in absolute terms, a very nice separation. As I mentioned earlier, we did not reach statistical significance, but the N was quite small. And we also saw a signal of reduced reinfection rate on the treatment arm versus placebo. And I should be clear, placebo was standard of care, and the treatment arm was standard of care plus placebo. So it was a pretty high standard that we were measuring ourselves against. And I'm just going to show you the pictures of 3 patients in that study that received drug. And so basically, the first 3 is one patient, the second 3 is the next patient, and the next 3 is a third patient. And you see the baseline picture for all 3. In these particular patients, each patient received 2 weeks of therapy, so 3 times a week for 2 weeks. And the second picture basically it was taken 2 weeks after the last treatment. And the third picture was taken 4 weeks after the last treatment. So you can see how the resetting the wound to a certain extent set a trajectory towards healing across the board in every single patient in this particular photograph. And we also saw patients in our study, even though the longest we followed was 7 weeks, that got to complete healing. So, again, we're quite encouraged by the findings from this small study. As I mentioned, we had a productive meeting with the FDA. We had a Type B meeting last week, and they have agreed to novel endpoints, which we believe are actually in our favor, that are very helpful to getting the results we need with a smaller sample size. We will also obviously be looking at the more traditional measures as well. And as I mentioned, based on our discussions with the FDA, if we choose to, we could actually have the next study be a registration or pivotal study. The commercial opportunity and all the indications we're looking at are quite significant. We've done some independent forecasting, and the diabetic foot ulcer chronic wound indication is in the U.S. alone about $1 billion opportunity. Discounted for discounts that we know that we'll have to give, so it's actually a realistic forecast. The orthopedic indication, which I've not spent too much time discussing, both with the treatment and the prevention indications, is also about a $900 million opportunity. The inhaled formulation for CF, and I'll spend just a minute more on that in a few minutes, is also a significant opportunity. It's about $500 million in the U.S. alone. And so as you then expand to a global opportunity, it becomes much more significant. I'm just focusing on the U.S. numbers right now. I just want to spend a quick minute on the cystic fibrosis indication. It's actually our inhaled formulation. We're looking at 2 orphan indications, both cystic fibrosis-related infections and non-tubercular mycobacterium infections. What we do know is that established biofilms, including Pseudomonas aeruginosa, are not adequately eradicated by current treatments. So we need something to work against the biofilms, which we do. We also know that there's now more than just a single pathogen in play in CF infections. It's historically been multi-drug resistant Pseudomonas, but there are many more pathogens in play. And so the fact that we have activity against some of these other pathogens is very important because it provides the right level of coverage both at the bacteria level and the biofilm level. We met with the Cystic Fibrosis Foundation about 2 years ago and showed them some of our data. They were a bit skeptical. They wanted to replicate it with their own researchers, which we gave them our drug to do so. The data came out very well. They then proceeded to say, okay, we will fund 5 studies. We have now completed those 5 studies, and those are what we call kill shot studies, that if something goes wrong, you don't move forward. And we succeeded in all 5 of those studies. So we have now gone on and developed a formulation, and we're in discussions with the Cystic Fibrosis Foundation where they would hopefully fund the future work, and we should know in the next 60 to 90 days whether we effectively will get a non-dilutive grant that will fund a good portion of our work going forward. So this is an area we're quite excited about. There's a company called Insmed. They just got approved recently for NTM, basically infections, with a black box because they cause pretty significant adverse events in the respiratory area. And their market capitalization is $1.3 billion today. So we do believe that there is a good business in antibiotics. I think we just have to do it right, and we believe we're following a roadmap that should allow us to not only bring benefits to patients, but also to make sure we do it in a way that works for investors. I'm not going to spend too much time on this, but we have a good IP position. In addition, we also have QIDP designation from the FDA for all of our indications, which means we would get extra exclusivity. So for our DFI, for example, if that's our first indication, independent of IP, in the US we would get 10 years of exclusivity. In Europe you'd get 10 years anyway. And for our orphan, which would be the NTM and the CF indication, we would get 7 years for orphan plus the 5 years of QIDP, so 12 years for all the orphan indications. So we feel we're in a very good position from an exclusivity perspective in addition to our IP. We have raised under $50 million to date. We are seeking additional investment to move our DFI and our pulmonary programs forward. But we're also looking at non-dilutive capital. We've actually raised a fair amount of non-dilutive capital. Our orthopedic study was funded in large part by the Department of Defense. We're looking at the pulmonary program hopefully being funded in a good part by the CF Foundation, and we're looking at other sources of non-dilutive funding through government sources, et cetera. So we want to balance dilutive capital also with non-dilutive capital. And in terms of getting us to the next level, the cost to do a pivotal stage study would be about $30 million to do that for DFI. To advance our pulmonary program to IND is about $7 million, and to do the first set of clinical studies would be $10 million. So if we had to raise money for all of those, including covering overhead, we'd be raising about $47 million. I won't get into detail, but we have a strong management team. I myself worked at Merck for many years, including heading up the infectious disease portfolio. From the commercial side of the business, I'm not a scientist. And we have a strong team that gives us the expertise we need in all the areas that we feel we need to succeed as a company. So I'll stop there, and I'd be glad to take any questions, either now or later. Any questions right now? I think everyone's very shy, but that's okay. Thanks very much.